Dear audience, an important condition for the successful mastering of the course of medical histology is the ability to correctly diagnose histological samples, to find and identify their microscopic elements, and to know the features of their structure and function. This lecture is dedicated to the development of these important diagnostic skills in relation to the topic nerve tissue. Nerve tissue is a group of tissues consisting of neurons or neurocytes, which are interconnected in the communication networks of the organism, and glial cells that perform a range of auxiliary functions to meet the vital needs of neurons. The embryonic origin of nerve tissue cells is neuroectoderm, with the exception of microglial cells, which develop from mesenchyme. The morphological classification of nerve tissue pays attention to cellular composition and functional determination of tissue elements. Studying the features of the microscopic structure of nerve tissue, let's start with neurons, the main cells of this tissue group, determining its specificity and role in the organism. Neurons or neurocytes are cells that are intended to generate and conduct excitation. They are integrated into communicative networks. Neurons include the nerve cell body and cell processes. Neurons are classified depending on the number of processes. Unipolar neurons have one cell process. Pseudounipolar neurons show a cell process that branches off from its cell body and then divides into two lines as the letter T. Bipolar neurons give rise to two cell processes branching off different cell poles. Multipolar neurons contain three or more cell processes. Multipolar neurons of the spinal cord. Large multipolar stellate-shaped neurons form clusters, nuclei, in the anterior horns of the spinal cord. A neuron has a body, perikaryon, covered with plasma lemma and processes, one of which is an axon and the others are dendrites. The perikaryon is externally covered with satellite oligodendrocytes, a type of oligodendroglia. The perikaryon contains a large, light-colored nucleus with a well-defined nucleolus. The cytoskeleton is well developed in the cytoplasm of a neuron. Its components are glued into bundles during fixation, stained with silver salts and revealed in the form of thin filaments, neurofibrils, which are artifacts of the histological processing of the samples. Chromatophilic substance in multipolar neurons of the spinal cord. The anterior horns of the spinal cord contain numerous multipolar neurons. When nerve cell bodies are stained by aniline dyes, one may detect basophilic clumps and grains of various size and shape. These structures are denoted as chromatophilic substance or Nissl bodies. Nissl bodies are also distinguished in sight where dendrites branch off, but they are never found in axons nor their tapering basements called axonal hillocks. Components of the cytoskeleton are glued into bundles during fixation and revealed in the form of thin filaments, neurofibrils. They are artifacts. The nuclei of neurocytes contain large nucleoli, which indicates high biosynthetic activity of the cells. Pseudounipolar neurons of the spinal ganglion. Nerve cells with a rounded perikaryon from which both processes, axon and dendrite, branch off the cell body together as a single process. At some distance from the perikaryon, the single process branches in a T-shape into dendritic and axonal processes. Therefore, such neurons are called pseudounipolar. The neuroglia is a complex of specialized cells that exert a number of accessory functions to ensure vital neuronal activity. The main functions of neuroglia include sustentacular, limiting and separating, metabolic, synthetic, locomotive, and protective functions. The number of glial cells is much greater than the number of neurons. The neuroglia is classified into macroglia and microglia, or glial macrophages. The types of macroglia are astroglia, oligodendroglia, and ependymal glia. Oligodendroglia, satellite cells and neurolemocytes. A section of a spinal ganglion fragment contains a cluster of pseudounipolar neurocytes and areas of longitudinally dissected myelinated nerve fibers. Satellite oligodendrocytes are flattened cells with oval or round nuclei that form a sheath around the perikaryon of neurocytes. Between the neurons there are interlayers of loose connective tissue with blood capillaries. Neurolemocytes or Schwann cells form the myelin sheath, which is part of the myelinated nerve fibers. Myelin sheath gaps are the boundaries between neurolemocytes. Astroglia. Astrocytes. Astrocytes are the most abundant glial cells of the central nervous system, which are characterized by numerous cytoplasmic processes radiating from the glial cell body. Astrocytes help to selectively move nutrients and other metabolites between neurons and capillaries. The elongated fibrous processes of astrocytes approach blood vessels and form extensions on them, vascular feet that completely surround capillaries, forming a perivascular glial boundary membrane. Some astrocytes also participate in perineuronal coat formation. Their processes terminate on nerve cell bodies. Microglia. Microglia are monocyte-derived cells of the central nervous system, less numerous than astrocytes, but nearly as common as neuron. Microglia are small cells located between neuronal cell bodies. Their short processes are also branched by secondary and tertiary processes, which gives the cells a spiny appearance. Phagocytic microgliocytes lose their processes and become rounded. Ependymal glia. Ependymal glia is type of macroglia lining the spinal canal and ventricles of the brain. Ependymal cells are epithelial-like cells that form a single layer lining the cerebral aqueduct. Ependymal cells contain numerous cilia on their surface, and their beats facilitate the transport of cerebrospinal fluid through the cerebral ventricular system. From the basal surface of some glial cells, long processes branch off, which extend inward and form the supportive apparatus of the brain. The main neuronal feature is to reflect environmental impacts by modifying electric tension on cell membranes. Thereby the cells conduct nerve impulses via their system of cell processes surrounded by glial cells denoted as nerve fibers. Nerve fibers are neuronal processes, or axis cylinders, surrounded by a sheath of sequentially arranged oligodendroglial cells in the peripheral nervous system, or by their processes in the central nervous system. In the peripheral nervous system, depending on the glial coat architecture, one may classify the fibers into non-myelinated or unmyelinated and myelinated nerve fibers. Glial cells of the peripheral nervous system that participate in the nerve fiber formation are called Schwann cells. Non-myelinated nerve fibers in the nerve ganglion. Non-myelinated nerve fibers are found in the form of longitudinally cut bundles lying between neurocytes as part of the sympathetic ganglion. Neurolemmacytes form oxyphilic cords in which oval nuclei lie at a slight distance from each other. Non-myelinated nerve fiber, ultrastructure. Non-myelinated nerve fiber is a structure consisting of several axis cylinders, neurocyte processes, immersed in the cytoplasm of sequentially arranged neurolemmacytes in duplicatures of their plasmalemma, denoted as mesaxin. The outside of the fiber is covered with a basement membrane. Each axis cylinder is surrounded by the plasmalemma of the neurolemmacyte. In the area of immersion of the axis cylinder into lemmacyte, the plasmalemma forms a duplicature, denoted as mesaxin. Some axis cylinders are not completely immersed in the lemmacyte and are separated from the space between fibers only by the basement membrane. Myelinated nerve fibers. Myelinated nerve fiber is a structure consisting of an axis cylinder, axon, surrounded by sequentially lying neurolemmacytes. Numerous windings of their plasmalemma around the axial cylinder form a myelin sheath. Along the entire length of the fiber in the myelin sheath, periodic interruptions called the myelin sheath gaps or nodes of Ranvier are occasionally detected. They do not contain myelin and correspond to the boundaries of adjacent neurolemmacytes. In the center of the fiber there is a light axis cylinder, a nerve cell process. The myelin sheath surrounds the axial cylinder. It has a high level of lipids, so upon impregnation with osmium salts it exhibits a homogeneous structure and intense black staining. The fiber is covered with a basement membrane on the outside, most clearly visible in the area of the Ranvier nodes. Myelinated nerve fibers in cross-section are represented by round-shaped structures. In the center of the fiber is a light axis cylinder. The myelin sheath surrounds the axial cylinder. Upon impregnation with osmium salts, it exhibits a homogeneous structure and intense black staining. Myelinated nerve fiber cross-section, ultrastructure, forming myelinated nerve fiber. In the center of the fiber there is an axis cylinder, axon. The axial cylinder is immersed in the cytoplasm of the Schwann cell. Each axis cylinder is surrounded by a long mesaxon, duplicature of lemma-side plasma lemma. Concentric windings of mesaxon around the axis cylinder form the myelin sheath. In the forming myelin fiber, mesaxon windings are few. Between them there are areas of the lemma-side cytoplasm. Formed myelinated nerve fiber. In the center of the fiber there is an axis cylinder, axon. The axis cylinder, the motor process of the neuron, is bounded by the plasma lemma and contains transversely cut elements of the cytoskeleton, transport vesicles and mitochondria. The plasma lemma forms a duplicature, denoted as mesaxon. The numerous oncentric windings of mesaxon around the axis cylinder are densely arranged and form a thick myelin sheath. Dear students, after examining the properties of nerve tissue, we have completed the series of lectures dedicated to the theoretical and practical study of the structure and function of the main types of tissues in our body. I hope that the fundamental knowledge acquired will assist you in mastering the core section of our discipline, the course of private histology. I wish you continued success in your studies. Stay with us for our next meetings on the HistoNavigator channel.